Thank you.
Welcome to GAAC. I'm here to introduce tonight's guest, who is Dr. Amy Williams. She was here last year, and she's an assistant professor of geology at the University of Florida and specializes in astrobiology and geology. Her research focuses on the formation and preservation of biosignatures, signs of life in extreme environments on Earth, which she uses to inform the search for signs of past life on Mars and elsewhere. Dr. Williams has been a member of NASA's Mars Curiosity rover science team since 2009 and contributed to the exploration of organic molecules on Mars. She's also a participating scientist at the Mars Perseverance rover mission, aiding in the search for ancient microbial life and the assessment of past habitability in Jezero crater. I hope I said that right. Yeah. In a recent study published in Nature, Dr. Williams and her colleagues reported the detection of diverse organic materials on Mars, suggesting a more complex organic geochemical cycle than they previously understood. These findings offer significant insight into Mars history and its potential to have supported life there.
Thank you.
Welcome, Amy.
Thank you.
That was awesome. Thank you, Lisa. Let me
And
go
she's
ahead
been a
and-
member of GAAC since 2025.
All right, excellent. Well, let me get my screen shared here. There we go. And can I confirm you can see it?
Yes.
It's loading. Now it's there. All right,
Yep,
excellent.
we can see on the call.
All right, cool. So yeah, thank you all so much for inviting me back. I am beyond thrilled to get to join you all again. And I thought this time I wanted to share with you sort of some exploration of what the evolution of the search, well, the evolution of the exploration of Mars. And of course, as an astrobiologist, I'm interested in how our search for life beyond Earth, especially on Mars, has evolved. And since we're now celebrating the 50th anniversary of the Viking lander missions, I wanted to do a little bit of a deep dive into how our exploration of Mars has evolved. And so this talk is a little bit more of like a almost like you're sitting in a classroom learning about stuff, but I'm hopeful that you will learn something new tonight that maybe you didn't know about Mars exploration over the years. So let me get this moving forward. Okay, so for our astronomers, of course, I have the image of Mars from the surface of the Earth. We've had all of these names for Mars over, the history of humanity, different names from the Egyptians, the Greeks, the Babylonians, the Chinese, the Romans. This planet, the red one, it's always appeared, red in the night sky and caught our attention over millennia. And so in 1610, Galileo was the first human to view Mars through a telescope. And the Italian astronomer Giovanni Cassini observed the southern polar ice cap of Mars and measured the length of the Martian day accurately to within 3 minutes. And this was done in 1666. So we started to get a little bit of a grasp of Mars as a world at that point. But in the centuries that have followed, others have tried to further explore Mars from having their feet on the Earth, of course, by sketching what they saw through ever-improving optics on telescopes. And actually, the surface of Mars had been completely mapped by 1841. So here's an example of one of those maps. The Italian astronomer, I always mispronounce it, and so I apologize to any Italians in the room, Schiaparelli, but I know I messed it up, was observing Mars and drew these long linear features that he saw on the surface, and he called them canali. This is Italian for channels or gullies. But this sort of bastardization of the term came to have it be mistranslated as canals. These features were actually eventually determined to be optical illusions, but this concept of canals on Mars actually took hold in our brains. And so, the Suez Canal had opened within kind of recent memory of these observations. and the work on the Panama Canal had just begun. And so the public imagination came to see canals as being sort of evidence for industry and civilization. This kind of launched our part of our, I would say, obsession with Mars as sort of being a habitable world. But the astronomer Percival Lowell actually took it the additional step and leaned into this, asking actually if Mars was inhabited. So he told the New York Times in 1907 that Mars is at present the abode of intelligent, constructive life. No other supposition is consonant with all the facts here. It sounds like if you say something confidently, people will believe you. So he set up what's now the Lowell Observatory in Arizona so he could study Mars. And he wrote three popular books about Mars at the time. Mars and its canals, and the one that really stands out to me, of course, Mars as the abode of life. So Lowell reasoned that Martians would have to have been peaceful given the cooperation required to build a worldwide series of canal networks. Speculation about what might be happening on Mars also fueled public imagination. And so Martians became sort of more common in popular culture. So in books, movies, radio plays, music, and advertising. And of course, Mars is also known as the bringer of war. And this brings about another way of thinking of these supposed Martians. Some representations saw these visitors from Mars as being benevolent. But another view posed Mars as a threat. So that theme spawned H.G. Wells's War of the Worlds, of course. And in that book, the Martians' own planet was drying up and dying. And so, in the quote from the book is written that they regarded this earth with envious eyes and slowly and surely drew their plans against us. I think many folks may recall that there was actually a radio play reading the War of the Worlds that people tuned into and actually thought Martians were invading New Jersey. And so this caught a lot of attention, of course, for like this misunderstanding. This was just a radio play, but this kind of concept that people could hear this report and think this is actually happening. There's Martians here. I think that kind of speaks to our expectations, or I guess our hopes for what Mars is, and sort of the symbol it serves humans without really representing what Mars really is as a planet. So moving away from sort of our perceptions of Mars to our observations, in 1956, this image was our best view of Mars. We had a Mars close approach in September of 56. The Mars was 35.1 million miles away. And since it would be at its closest and brightest, our astronomers trained their telescopes on Mars. And so this was the best image we could get from the 100 inch telescope at the Mount Wilson Observatory in Southern California. So just to give us a sense for like we were, what, 15 years-ish, 13 years from landing people on the moon, and this was the best version of seeing Mars that we had. So of course, things were about to change dramatically in those next 15 to 20 years. This is actually the global mosaic of Mars that the Viking orbiters were able to collect. So we're about to get a whole new perspective on Mars. So let's jump into sort of the modern space-based exploration of Mars. The Mariner Mars program is part of this, and so between 62 and 73, NASA built these 10 flyby probes meant to explore several rocky bodies, Venus, Mars, and Mercury for the first times. So in November of 64, NASA launched the Mariner 3 and 4 probes. Mariner 3 failed at launch, but Mariner 4 successfully launched and sent back these first flyby images of Mars. The photos showed craters and nothing else. No canals, no Martians, no life. This was called a scientifically startling fact by the Mariner imaging team at the time. So in 69, NASA sent Mariner 6 and 7, and that mission photographed about 20% of the planet's surface, including the dark features that were long seen from Earth and interpreted as the canali, or the canals. And then finally in 71, we sent Mariner 9 is the first Mars orbiter, which imaged then 85% of the surface. So within a relatively, within a decade, basically, we really increased our observations of Mars and our understanding of it as a world. Now, unfortunately, that understanding did not sync with what we had sort of built as this, shared public concept of what Mars might be. So I've got this article a colleague of mine from Australia sent. He found this in his mom's attic from a newspaper from 1969. It says Mars shown as grim, dead planet. And so a quote from this article says that scientists today saw Mars's equatorial region as a grim, crater-pitted, moon-like planet with no evidence of water or cloud to support life. So this is a pretty, bleak view of a world that we once thought might not only have been habitable, but be inhabited. Um, so this sort of sets the stage for our next step with the exploration of Mars. But first, what I want to do is kind of point out to you, sort of starting from the initial landed missions that we're looking at to the things that are actually operating today or are going to be heading to Mars soon. And I wanted to give you a sense from the orbital missions that I've talked about, or the flyby missions, and we'll talk more about some of the other closer images we were able to collect from Mars before we landed. This gives you a sense for where we've landed. These represent missions from different space agencies, of course. It's not just NASA. But over, what the past 50 years, we've actually made pretty good progress in peppering the surface with some successful missions and then some, unfortunately, that made craters. And so I do want to point out though that it's not just landed missions, of course. And if you have a phone on you and if you want to do this, you can use your phone to take a picture of this black and white QR code at the bottom, which will give you a permanent link, or should give you a permanent link to this figure, which I think is really cool because it actually shows you the different Mars missions. It's a little bit dated. It still has ExoMars at 2022. ExoMars is meant to launch now in 28. But it shows you the different missions, those that were flybys, orbiters, and landers, and then rovers, and then which nation they came from and whether they were successfully landed and operated or not. But I think it's just a really cool way to kind of get a sense for how many missions Earth has sent to Mars. I'll give you all one more second in case you want to use your phones, but I can also bring this up at the end if you're interested. Okay, so I want to jump into the very first attempts to land on Mars, and this starts with Mars 2, 3, and 6. So these were actually missions from the Soviet Union. In 1970, Soviet Union began the design of the Mars 4 and Mars 5 NM missions. That second mission was actually a Mars sample return mission, which I know you all have heard me speak about previously, as was planned by NASA and ESA. So both of these projects were later canceled. Again, another trend that we continue to see through the lifetime of space agencies and their operations on Earth. But in 71, the Soviet Union stepped back a little bit and sent two probes meant to land on Mars and carry landers with them. So Mars 2 failed to land and actually impacted Mars, created a little crater. And the Mars 3 lander actually became the first probe to successfully soft land on Mars. Transmission stopped after not quite 15 seconds. They don't really know why, but each probe actually contained a Mars rover that never deployed from the vehicle. In 73, the Soviet Union sent two more landers, so Mars 6 and 7, trying again. And Mars 6 transmitted data during descent, but then it failed before landing. And then Mars 7 actually, unfortunately, prematurely separated from the carrying vehicle and missed the planet by 800 miles. So close, but not quite. So this is sort of where we're coming from, and NASA answers these missions a little bit with some of the Mariner missions, taking images of flybys and orbiting, but the real big step forward was with Viking 1 and 2. So it's so funny. I have this caption here saying Carl Sagan standing with a replica of a Viking lander because I've actually run into situations teaching in the classroom when students don't know who Carl Sagan is. And so I'm like, I know, yes, Patty's face says everything here. Um, so I'm like, I tell you what, we're gonna pause and talk about Carl Sagan. So I'm still sharing with these kids who this person is. Of course, Carl was a big proponent of the Viking mission. Viking was so it's two orbiters carrying two landers, and the mission had three goals: to get high resolution images of Mars, to characterize the structure and composition of both the Martian atmosphere during descent, as well as the surface of Mars, and then, no big deal, to search for evidence of life on Mars. I frame it in this way, and I'm silly about it just because, as it turns out, I think when Viking launched, I'm not sure that we knew that there were hydrothermal chimneys at the bottom of the ocean that supported a chemosynthetic energy pathway for life on Earth in a way that was different from anything we'd seen on the surface. And so we really, I keep saying that we were learning to search for life on Earth at the same time we were trying to search for life on Mars. And I think that really plays into the story of Viking because the search for evidence of life with these experiments on board the rover were inconclusive. So let me show you some of the really cool things that we did find, though. We collected these. I say we. I wasn't around, but NASA, the Viking team, collected these high resolution images of the Martian surface, and many of these images actually showed evidence of water that had been flowing on the surface of Mars a very long time ago. So branched channels that, on Earth at least, formed due to rain, these teardrop-shaped islands that are carved by flood waters, additional branch channels over here in Margaritifer sinus quadrangle. So we're seeing evidence for water for the first time, which were missed in the Mariner images. I would be remiss to not show the famous or infamous Face on Mars, which I remember some, silly infomercial as a kid being like, what's NASA not telling us? Now, since I've worked for NASA, I can tell you they're telling us what they saw. It was just this plateau. But this is the kind of thing where higher resolution images can make the world of difference in understanding and basically an entire other world. So let me get to the sort of the pinnacle of the Viking work here. Viking carried a biology experiment. Again, I say, we were learning how to identify life on Earth while searching for life on Mars. And so I'm trying to bring this very high level, but step you through what the four experiments on board the biology experiment were. So the first one was meant to analyze basically untreated Martian soil by heating it up. And this is actually a technique that we use on the Curiosity rover now, and something that I've used on that mission. Where Lisa introduced me and said that, I had a paper recently where we found evidence for organic carbon on Mars. This experiment with Viking did not detect organic carbon. That was very puzzling. So even though organic carbon wasn't detected, the rest of the experiments proceeded. So one was called the gas exchange experiment, where there was some exchange. We saw oxygen disappear when you mixed a nutrient solution with the soil, and some CO2 evolved. So that was interesting and maybe confusing. The one that really sticks with everybody, though, is the labeled release experiment, the LR. And so what this was that we took a scoop of Martian soil and we added this radiogenically labeled C14 nutrient solution to the soil. And so if you monitor the gas that comes off of there, if some of it has that carbon 14 incorporated into the carbon dioxide, you might use that as evidence for microorganisms that are actually metabolizing the carbon that you gave them in that nutrient solution. We did see CO2 evolve, and we were trying to understand why that was the case. So this is why this particular experiment was inconclusive, because the way it was designed and the way we expected things to work, that would have been indicative of an organism metabolizing the carbon. However, something we didn't know about Mars at the time was that it had what we call superoxide salts. So these are like the perchlorates that we've detected on Mars. When you have UV light, which we know the surface of Mars is bathed in, mixed with carbon dioxide and regolith, and it reacts with those perchlorates, you can actually get these little organic molecules that form from that. And they can be destroyed actually in the top experiment here. So things were just inconclusive with these experiments. The last one, pyrolytic release, also saw a little bit of organic matter being generated, and why that happened remained inconclusive. So, most of us now say, no, Viking experiments did not detect evidence for life. And I would say, like, yeah, it's nothing is conclusive about this. And so this was done in the mid 70s, and we actually had to wait for quite a while for sort of a rebirth of Mars exploration. And part of the reason this came about was actually a news report that came out in the mid 90s about potential biosignature or evidence for life in the Allen Hills meteorite. And so this was Dave McKay's paper from 1996. This is a meteorite that was collected from Antarctica in 1984. The meteorite itself is 4.5 billion years old. That's the age of the Earth and probably the age of Mars. So it is extraordinarily old. And I don't recall if we know when it was ejected from, I mean, we must know when it was ejected from Mars, I would think, to know how long it orbited in the solar system. But we know it arrived on Earth about 13,000 years ago. And so the McKay paper really did put Mars exploration on the front burner. The paper argued that the detection of organic carbon, these PAHs or polyaromatic hydrocarbons, the chemistry and mineralogy of some of the minerals they saw in the rock, minerals called carbonates, the formation of other minerals like magnetite and iron sulfides, and these interesting surface features like the famous sort of like worm image that you see here on the top left. All of that came together for them to argue that they think that there was a biogenic origin for some of the observations in this meteorite. And this really launched what I do now, astrobiology. It was called exobiology at the time. Um, Basically, everyone except maybe the team that worked with Dave McKay agrees that there's not evidence for life in this meteorite. But what it did do was spark the conversation. What does it take to confidently identify a biosignature? What's reasonable? What's not? Why is that the case? And so the funding for Mars exploration actually increased again once we had this discovery, and all of the scientific dialogue that went along with basically us arguing, yes, it is, no, it isn't, over this meteorite. So, Mars exploration kicks off again in situ on Mars with the Pathfinder mission. Pathfinder landed in 97 in this ancient floodplain in Mars's northern hemisphere, and it carried the sort of demonstration rover Sojourner with it. This was the first successful Mars rover. It traveled a few meters around the landing site and explored the rocks in the area. And this is really sort of what launches our ability to build rovers to explore a surface of Mars. This little rover and the lander, Pathfinder lander, demonstrated that there was that past Mars was warmer and wetter than it is today, and that there was definitely liquid water and a thicker atmosphere. So this sort of complements what the Viking orbiters, the images they sent back. So, and these are just like little snapshots of what Mars exploration looks like. So I don't get to spend too much time on each mission, but you're sort of seeing this evolution of our exploration. We had a couple of bad years where ESA's Beagle 2, ESA's Schiaparelli, and NASA's Mars Polar Lander all failed for various reasons and did not deploy. Now, Beagle 2 was actually a life detection mission as well. So we're still trying to build missions that can look for evidence of ancient or even extant life on Mars. And so Sojourner, though, really paves the way for us to rove on Mars, start to follow the water, look for evidence of water on Mars with the Mars exploration rovers, Spirit and Opportunity, to look for habitable environments, places where life would want to live if it were present on Mars with Curiosity, and finally to search for ancient life in with the Perseverance mission. And so I'm going to spend a couple of slides just stepping through some of the cool things that these missions have found, and then we'll kind of get into what's next. So let me start with the Mars exploration rovers Spirit and Opportunity. Let me see if I can get this video to play for you all. Here we go, hopefully. We'll see if it wants to work. Maybe it won't. There we go. Then the twin rovers, Spirit and Opportunity, were sent in '03 and '04. Spirit landed in Gusev crater, which was thought to be a crater lake, and Opportunity landed in Meridiani Planum, where there's a lot of the mineral hematite, which is evidence for past water. And both missions were built as twins so that they can perform the same experiments. The video I'm showing you here is actually showing how, and I hope it's rendering for you all in the room as well, but it's showing how these vehicles had to land with these big airbags. So when they landed, they bounced way up in the air and bounce until you finally lose your momentum and the airbags deflate and the vehicle is able to drive out. And so I'll just pause this here. These missions were actually able to determine that both of these locations were drenched in water in the past, and this was the first strong direct evidence for liquid water on Mars at some time in its past. So here's an actual image of when the airbags deflate and the vehicle is able to drive off of the lander. So these missions were designed for 3-month operation. And so that's part of why the first 3 months of these Mars missions, we live on Mars time. Initially, they thought they only had that amount of time to operate them. But Spirit ended up operating for 74 months. And Opportunity continued on for, what do I have here? 173 months. It was only lost in June of 2018. It basically ran a marathon, surpassing 28 miles on its odometer. And these missions provided sort of long-term detections for evidence of ancient water and ancient environments on Mars in a way that we had no precedent for prior to these incredible missions. And these really were harbingers for the next generation of rover missions. So before we get into rovers, let me pause for a second and talk about the Phoenix polar mission, because that actually ends up being a way to tie back some of the results we saw from Viking. So Phoenix landed in 2008. It only operated for a couple of months because it was so far north that in the winter it wouldn't get enough sunlight to charge its batteries and run its survival heaters. It was used to assess the local habitability potential and history of water on Mars in the poles. So this was the first successful polar landing mission, and it detected water ice for the first time with a landed mission. And the really big thing for the Viking missions is that it confirmed the presence of those superoxide salts, which likely were the reason the reactions occurred that did with the Viking experiments. I really love this image because this is showing sort of the water ice detection that the lander made. It used its scoop, dug into the regolith, and from the little image that you see for Sol 20, so basically day 20, you see some little white particles in the scoops area here, and 4 days later, they're gone. And so the rate at which they sublimated tells us that they were water ice and not CO2 ice. So this was like our first true evidence that there's still water on Mars. It's just frozen. It's a lot less than on Earth, but it's still there. So this actually comes into play when we talk about future missions to Mars to look for extant life. So we leave Phoenix behind at this point and go on to a few missions you have heard me speak about before. I'll give you just my favorite highlights for Curiosity and Perseverance. But Curiosity is sort of that next step, sending basically a roving chemistry laboratory to Mars. The overall objective for Curiosity is to study Mars's habitability, including its climate and geology, and collect data like radiation data for potential future crewed missions to Mars. We actually just celebrated our 14th landing versary, is what we call it. So 14 years of being operating continuously on Mars with Curiosity. And you've seen some of these images before, but we have evidence for abundant water in this location we're exploring called Gale Crater. We know that in the past, it kind of looked like the middle left image you see here, where there were these ephemeral lakes that had relatively long lifespans over sort of the course of all of these lakes forming and filling and drying and filling. Um, we've found organic carbon in several of the locations we've explored with Curiosity. And we've been collecting radiation data that helps us to understand how humans might be able to navigate the surface of Mars if we ever are able to send crewed missions. This is one of my favorite images of Curiosity exploring Gale Crater, and this was before we even started climbing the mountain. This was our very first drill hole at a place called John Klein. And what's really awesome is that once we drilled into the subsurface, 6 centimeters down, which isn't a lot, but was the deepest we'd ever drilled into the subsurface, we found that the red planet is just a veneer. It's only millimeters thick. Once we drilled down into the subsurface, even 6 centimeters, that gray rock that you see, that shows us that there are minerals in different, what we call redox states. So some are oxidized, making them red, some are reduced, keeping them gray. And this is actually really helpful for establishing what we call habitability. So whether life like our bacteria would want to live there. In this environment alone, we were able to determine that there was water in the past. It was a lake bed. There was organic carbon present and preserved in these rocks, and that the types of minerals found in both the red and the gray rocks actually are the kinds that bacteria on Earth use to generate their own metabolisms or to power their metabolisms. So within 7 months of landing on Mars, we actually had characterized our first of many habitable environments in Gale Crater. And so I wanted to use this opportunity to tell you some of the more recent findings that have come out of the Curiosity mission. And I know it's organic chemistry, and I know it's 8 whatever at night, 8:40. But if you stick with me for a second, I'll show you some really cool stuff that's been coming out of the mission recently. The first is this paper from my colleague Caroline Frisney, and you'll see I'm a co-author on this paper. This is called Long Chain Alkanes Preserved in a Martian Mudstone. So why is this important, and why do we care? Seeing these kinds of hydrocarbons is really cool because these are the longest, I think still to date, the largest organic molecules we've found on Mars. What's really intriguing about this is that when you see like when you do an experiment on Earth to sort of mimic what we did with Curiosity rover, you don't actually get these molecules coming out of the mass spectrometer. What you actually have to do is take a precursor molecule, what we call a fatty acid, and that's what I'm showing on the bottom right here. You see that the shape of the molecule is different from the alkane, and the alkane is what we see, these 3 alkanes in the rock. So what it means is that these molecules were almost certainly these precursor molecules in the rock. And then our experiment actually broke them down and turned them into these hydrocarbons, these alkanes. Now, this is really intriguing because if you look at the sort of middle right image here, it says biotic and abiotic. When you look at what rains down from meteorites on Mars, we do see organic carbon, we do see fatty acids, and we do see alkanes. What you don't see, though, are any fatty acids from meteorites that are longer than 12 carbons long. So that's where you see the orange and the blue overlapping here at the number 12. That's sort of the boundary where you don't see anything longer than 12 carbons long. If we have a carbon 12 alkane coming out of this experiment, like I'm showing you here, that means it came from a fatty acid that had to have 13 carbons. I don't know how you do that without life. So I'm not saying that this is evidence for life, but I'm saying I don't have a good explanation for how you make something any longer than 12 carbons long on a meteorite. So how in the world did this fatty acid form on Mars? So that's a little bit of organic chemistry for you. There's been some follow-up studies, or I guess arguments or modeling that's been done. Some of the co-authors from the paper actually worked on a separate paper that has a very leading title. Does the measured abundance suggest a biological origin for the ancient alkanes preserved in that mudstone? And so some of the co-authors are exploring, like, if you have this kind of organic carbon preserved over billions of years, how much had to originally be there for this amount to still be preserved? And they were arguing it actually seems like it might be a lot. And how do you generate that much organic carbon? Normally, I mean, like on Earth, it would be from life. So it just gives us a lot of questions to try to understand how the search for life on Mars is evolving. It's not Martians walking up to us, not little green men, but it's these little hints here and there. And so I just wanted to show you, Lisa kindly commented on a paper that I had come out earlier this year, and I wanted to share that with you all as well. The paper came out in Nature Communications, and it was documenting an experiment that I was able to lead with a certain chemical we carried on board that was meant to react with the rock, and it did. And what it demonstrated to us was that you see this big complex organic clump of stuff in this figure in the center bottom. Um, this is an example of what we think some of the organic carbon might have looked like in the rock before we broke it down with the experiment that we performed, this what's called this TMAH experiment. And so what this tells us is that there's ancient, sort of more complex organic carbon preserved in Martian bedrock, despite it being there for probably 3.5 or more billion years. It's preserved, it's detectable, and it gives me hope that if there is something made by life, that we might actually have a chance of detecting it. It wouldn't be beyond our ability to see it. So this is a paper that I had come out recently, and I wanted to share it with you all. And so I'm going to move on sort of to our last flagship mission right now, which kind of builds on this story of searching for an environment where life would want to live and have the components that that life needs to have survived in the past. And that is with the Perseverance mission, which I know I've had the opportunity to share with you all previously. Just as a reminder, though, why do we go to Mars to look at rocks? When you look at the oldest rocks on Earth, most of them are younger than 2.5 billion years old. There's only a couple of little pockets around the world where we still have rocks that are more than 3.5 billion years old. But more than 50% of Mars's surface is 3.5 billion years old or older because Mars doesn't have plate tectonics. Its rocks haven't been recycled the way that they have on Earth. And so Mars offers this unique window into what basically a planet looks like in its infancy, what those rocks look like without plate tectonics. And so Perseverance landed in Jezero Crater. One of the things we are interested in exploring is you actually see it in this beautiful image from the HiRISE orbiter, and it's the river delta that flowed into the lake that was once in this crater. The HiRISE orbiter actually took this, or the camera took this incredible image of Perseverance, and that's the little white dot at the bottom of this super grainy zoomed in image, and our supersonic parachute deployed as we're landing in Jezero Crater. So this really cool opportunity to use multiple vehicles to sort of monitor each other. So I think I shared this with you before, but I think that one of the other cool things about using multiple cameras to document our exploration of Mars is that in this case, we used basically some GoPros on a $2.5 billion rover and got some of the most incredible images of Mars that we can, or videos of Mars that I think we can possibly hope for. The left is our supersonic parachute deploying. The center is actually the descent stage, and there's actually, like, if this was on Earth, you would see some flames, but we're on Mars, so you actually don't see it, as the vehicle descends to the surface of Mars, and then the rover actually drops down on this tether, and that's what you see on the far right, as the vehicle lands, and then the sky crane, which is that sort of rocket backpack, flies off. So anyway, this is what it's like to land on Mars. And I know last year I was able to speak with you about sort of what Perseverance is exploring. And so I'm kind of cutting to the meat of it here. We're at 27 rocks that we've collected, two samples of the soil, or what we call regolith, a sample of the atmosphere, and then three witness tubes. So these are basically clean tubes that we just seal at different points in the mission so that we can see what our contamination is like. So that if we ever do get these samples back, we can actually say something about whether there was any contamination present, or if what's in the sample tubes is pristine. And so I've got this sort of family portrait of our samples on the left-hand side here, showing the where we kind of grind into the rock to take a look at the rock, what the core actually looks like when it's in the tube, and then what it looks like once we seal it. And so that's what you're looking at with the family portrait of samples on the left-hand side. The right-hand side is showing you where samples up to at least sample 30 were collected. And so what I want to do is take one more moment to tell you more about the sort of the evolution of the search for life on Mars here by zooming in on this region where sample 28 was collected. So we are looking with Perseverance. We're using a different technique than on Curiosity to look for organic carbon. We're using an instrument called Sherlock, which is using basically Raman spectroscopy, so like light reflecting and reacting with vibrational modes in organic carbon in order to tell us something about the nature of that organic material. And so when we zoom into the location that I showed you on the previous map, we're going to zoom in on an area that's called Bright Angel. And you can see it's sort of this white, light-toned material in this river channel that is basically leading out of Jezero crater. So you can kind of see it from orbit in the top image, and then what it looks like from the rover looking at the landscape in the middle here. When I go to the next image, here we go. Here is a zoom of where we actually collected the rock sample and sort of what this outcrop looks like. And when we zoom in on it a little bit more, we see these, what we call leopard spots. Not made by leopards, of course, but they have this sort of ring of light-toned minerals and then a dark tone in the center. And so the combination both of the types of minerals that make up these leopard spots and the detection of organic carbon with that Sherlock instrument on board Perseverance was actually used in several papers to argue that there's what we saw with Curiosity, this complex, what we call macromolecular organic carbon present in the rock. And so the minerals and the organic carbon, all of that together was used to report the first detection of a potential biosignature on Mars. So what's a potential biosignature? It's something that could have a biologic origin. If you saw it on Earth, you would say, yeah, life makes that. But we need to further study it before coming to a decisive, definitive conclusion about whether it was made by life. So it's basically giving yourself an out if you're wrong, but it's so compelling to us that we really want to study it more. This is the sample I want to have returned with Mars sample return. And I find all of this so intriguing to show you the stories across Curiosity and Perseverance because I just showed you that we found evidence for complex organic carbon with Curiosity. So we're actually seeing these diverse organics results are consistent across two missions using two different instruments in two different locations. I think this means that we have a lot more to look forward to with regards to habitable environments on Mars, the detection of a far more robust, organic carbon cycle on Mars. And I do have hope that if any of this organic carbon was made by life, that we might actually be able to make that detection even with these vehicles. But of course, the more we learn on Mars, we can build better instruments to address questions as we learn more and more. Once we knew there were superoxides on Mars, we could come up with experiments that get around what it does to organic carbon and kind of changing our experiments. And so that leads me to basically my last slide here, which is what's next in our search for life on Mars. The Rosalind Franklin ExoMars rover is launching in 2028. Thankfully, it has a launch vehicle again. This mission is going to be able to drill 2 meters into the subsurface of Mars, unprecedented depth. This is going to give us the opportunity to search not only for evidence of extinct life, the way that Perseverance is searching, but if there was extant, basically recent or even present life, this vehicle has some capabilities to detect that. It has an instrument suite that's sort of the next generation of the organic carbon detector that I work with Curiosity. So that is a mission that's actually built and launching. And so then what's next beyond that? I had the opportunity to lead this study of what's called the Mars Life Explorer mission, which has actually been further studied by NASA as what they're calling the Search for Life mission. They love the acronyms, so they can run with it if they want to. Um, but Mars Life Explorer is actually a lander concept I helped to lead that would allow us to drill 2 meters into the subsurface where there is water ice. So not at the pole necessarily, but moving up into the northern latitudes, to search for evidence of extant life in that mid-latitude ice. If there's a place where we're going to have um, the potential for life to still be on Mars, there's a couple of different places to look. The subsurface is one, and places where there's water ice is another. And so this kind of combines the two of them. So this is my, my school lecture for you all on a Friday night. What the evolution of Mars exploration has looked like. And this has helped me to really appreciate the work that went into especially the Viking missions, but really all of the exploration that we do in space. So I would say that I am optimistic for the future. I do, I know Rosalind Franklin is launching. I'm hopeful that we will one day get samples back from Mars that we can study. And I think we have so many compelling samples already with Perseverance. Um, I think that there's a decent chance that we might be able to say we found evidence for extinct life on Mars, maybe in my lifetime. We'll see. I'm going to be optimistic and say it's possible. So with that, I want to thank you all so much for sitting through this. I think I ran a bit longer than I intended to, but thank you for writing this out with me. I'm going to stop sharing so I can see you all and hopefully chat with you. Thanks.
Hey.
Yay. Sorry that ran a little
Thank
late,
you.
you all. Thank you so much, though. So last time I think I took some questions. Michael, I'll defer to you. I know it's a little late right now, but yeah, let me know if you all have questions.
That's fine. Can you hear me all right?
Yes.
Okay, well, looks like online, Carl has a question, so.
Yeah, Carl.
Thank you. What instrument would you like to have on Mars, and what would you do with it, and where?
Um, I would like to have, so instead of the instrument, can I have like an ability? Can I
Sure.
have, yeah, can I have the ability to send basically a full operating wet chemistry lab? That that would be ideal, or to bring back enough material from Mars that I can do the experiments that I do on Earth normally with enough material to actually get some organic carbon out of the rocks. On Mars, I think the organic carbon is going to be so much less concentrated than it is on Earth, and that's what really want to get at. I want that material to be able to say something about its origin, whether it's from life or not. So I think that would be sort of my like ideal scenario. Something I'd be very excited to see though would be a scanning electron microscope. So the ability to see basically you could see cells with an SEM. And so being able to do that in situ on Mars, I think would be an awesome opportunity to not only characterize the material, but to maybe even look for cells.
The benchtop ones are getting pretty small. Would anyone ever put one on a rocket and send one out?
So actually, I worked with a team that was trying to develop a variable pressure SEM that could actually operate with a CO2 atmosphere background. So there is a push to actually it's miniaturized, but then to also make it space flight hardy. So yeah, there is a push in the instrument development community to do this.
Cool. Thank you.
Yes, thanks.
All right, Alan?
Yes, thanks. Great presentation. You probably talked about this. I had to leave the room for a second, but what is the recovery mission for those samples? When is it scheduled for, or is it even built yet?
No, so yeah, that was the Mars sample return architecture. It is currently no longer funded. But I'd said just at the beginning of the talk that I am optimistic that some version of this may be reborn in the
Great.
coming years. So no, there is some development that has occurred though with regards to how to collect the samples, how to launch them, how to retrieve them. And so there's some sort of pretty advanced scaled tests of this at the Jet Propulsion Lab in California right now. All of that, of course, has been paused when the funding was pulled, but some of this has really moved into more mature versions. So I am going to remain optimistic because I don't like the alternative.
I'm assuming those samples can stay there for a billion years and still the technology and whatnot will be there to pick them up and
Yeah, I think so long as something, if we can pick them up now, which we can, then I think that any technological evolution will just enhance that.
sure.
And I think, yeah, so and the samples are hermetically sealed, and we looked at how the sand is moving for the samples that are depot
Mm,
on the ground, and they're not going to get buried. I mean, in a billion years, yeah, they might get buried, but
sure.
we will not be here to collect them. So I'm less worried about that. And then the other samples are on board the rover right now. And so I guess the biggest thing would be if we know the rover is reaching sort of its technological limits, then we might need to dump the samples. So that's the other concern to think about down the road. But like I said, I'm hopeful that we'll maybe have a new architecture to bring these samples home.
Thanks. And do we know who, or do you know who Rosalind Park or Rosalind Franklin was that
I
they
know,
named?
yes, yes. So I'm thrilled to have a mission, number one, named after a female scientist,
Mm,
who's like leading us to think more about DNA and sort of the fingerprints for life. Yeah, so
thank
I'm
you.
thrilled to have that.
Great presentation. Thanks.
Thank you.
All right, thank you. Arthur, what's your question?
Of course, I've got a question. First, we know who to thank or blame for the defunding. Maybe we'll send Elon to pick up the samples personally. The other thing is, I'm just wondering about, I know the atmosphere is almost a vacuum on Mars. And I'm wondering at the bottom of the continent size copy of the Grand Canyon there, the Mars
Valles Marineris, yeah.
barrels. Yeah, I wonder if the atmospheric pressure in it down at the bottom of that is high enough that things could have stuck around longer than in the more vacuum-oriented upper levels above ground. I mean, I don't know how deep that is. It's probably several miles deep,
It is,
and
yeah.
it's got to be a higher pressure down there than it is above. Like,
Yeah, it's not, yeah, so it's not a vacuum. It's certainly far, far, far lower than on Earth.
of course, yeah.
But there are definitely microorganisms on Earth that are able to survive at those pressures. And
The
so,
tardigrades, yeah, the
oh, and
tardigrades.
yeah, I mean, those are way more advanced even than like bacteria. So yeah, it is so I would be less concerned about, whether we'd have to go to the bottom of Valles Marineris in order to collect this or to look for that evidence of life. I think if an organism was on Mars, it would have evolved to deal with the lower pressure because it would have had a geologic time basically to evolve. So if it could have survived, it could have figured out how to deal with the lower pressure. But that is a really good point that, several miles down, there's going to be slightly different atmospheric pressure at the bottom of Valles Marineris. And there are some cool mission concepts that are meant to go into Valles Marineris and explore the incredible geology that's exposed in the side of the rock wall there. Who knows what we would find at the bottom? That's a really good point.
I'm sure some things don't sublime as quickly down at the bottom as they do on the, I studied vacuum for years. I worked
Hmm.
in an industry with high vacuum, -9 to 12 scale. And I'm pretty sure that it would be better lower to look for anything that needs a little more pressure to stick around.
Yep. Yep. Nope. I agree with that.
Hey, thank you very much. That was a great lecture. And
Thank
thank you,
you.
Michael. Have a great night.
All right, do you have any questions in the room?
Yeah.
Yeah, your microphone's over here, so speak up.
Okay. How do you define biosignatures? I've read a little bit about methanogens, and that sounds to me like it's a whole different concept of what could be considered, quote, life.
Okay, yeah. So yeah, methanogens are microorganisms that methanogens are going to make methane. Methanotrophs are going to eat methane. So yeah, that is just a metabolism that some microorganisms use to generate energy. When I talk about a biosignature, that is anything that can tell us it was made by life. You want it to be clear enough that you can say you couldn't have done this abiotically or without biology. And that's where we have sort of those distinctions between a potential biosignature, which is something we need to study more, and then what we call a definitive biosignature, which is if a Martian walked up to you kind of deal, right? Or if you found a dinosaur bone on Mars, you would have a lot of explaining to do to try to come up with a reason why that was abiotic and not biologic. So that's what a biosignature is, anything that had to be made by life. I hope that addresses your question.
All right, any other questions in the room? Anybody? On this side? On that side? Any more online? Going once? Thank you, Amy, that was terrific.
Thank you all
Amy.
so
Thank you
much.
very much, Amy. And
I
thank
really
you, Michael.
appreciate
It's
it.
always a good time.
Excellent.
Yeah, thanks, Michael.
We'll see you
Night.
next spring.
Have a great weekend. Thank you very much.
Thanks, Mike.
Wonderful.
Thank
You all
Did we
take
find
care.
did we find any
Bye.
volunteer to go help the dancers?
I couldn't go dance.
Michael? Is Michael gone?
Yes,
Still there.
Dick is going, he's left already.
Awesome.
Great.
Great.
Peggy, have a great
All right.
weekend.
Good night. Bye, everybody.
Good night,
Good
everybody.
night, folks.
Good night.
Closing
Bye,
the
Dennis.
doors.
Bye, Dennis.
Thank you
Bye,
again.
Ann.
Bye.
Night,
Bye,
everybody.
Michael. Thank you.
You.